Well, let's get started. We're going to start by looking at nucleic acid. So during my little walkthrough, I'll give you some tips and tricks and just reminders on things that you need to know. So for nucleic acid, please make sure you know how to draw the structure of a nucleotide and be able to label. So over on the empty space here, let's draw a nucleotide together. Okay, so any nucleotide consists of three major parts. So we have the ribose sugar, this little circle on the left is the phosphate group, and on the right-hand side here, we have the nitrogenous bases. We're going to add a bit more detail to it. So there are two types of ribose sugar, which makes up different types of nucleotide. If the ribose sugar is just ribose sugar, then it's the base for RNA, or the type of sugar for RNA. But if it's deoxyribose, meaning it has less oxygen, deoxyribose, then that will be the type of sugar for DNA. Okay, so nothing to add for phosphate group. All phosphate groups are the same, but you know there are different versions of nitrogenous bases. So if you're looking at A/T/C/G, these are the four bases for DNA molecules. And then if we replace T with U, then we're talking about the bases for RNA molecules. Okay, now just a reminder, a lot of people get this confused. The structure that we just draw is a nucleotide. So these are the single blocks that make up a larger molecule, such as DNA and RNA. So we are talking about monomers. And you know, mono means one. When multiple nucleotides join together, then you have a chemical molecule called nucleic acid. So nucleic acid is many nucleotides linked together. So in here, we are talking about polymers. So poly means many. So please make sure you are able to distinguish between a nucleotide and a nucleic acid. And you should be able to recognize that both DNA and RNA are types of nucleic acid. Okay, so like what it said here, two types of nucleic acid. Oops, don't want to move it. Bit tricky to highlight it, so I reckon I'll just underline it. So two types of nucleic acid, DNA and RNA. So you don't need to worry about the full names of the two molecules. What's more important is the distinguishment between the two. Okay, so rule of thumb, DNA is a double-stranded molecule. So if you're given any diagram and the diagram asks you which kind of molecule is that or which type of nucleic acid is that, the thing that should give you the most clue is whether the molecule is double-stranded or single-stranded. If it's a double-stranded molecule, it has to be DNA, and if it's single-stranded, then you're probably looking at an RNA. Okay, so here are the functions and descriptions of the two types of nucleic acid. So DNA contains the code for protein. It consists of the four nitrogenous bases, A, T, C, and G. It's double-stranded, and the type of sugar that is a part of the molecule is deoxyribose sugar. On the other hand, RNA has the function of reading the code of DNA and carry the code to produce proteins. Okay, so pretty much RNA is a copy of the DNA, and it's able to leave the nucleus and go into the cytosol, whereas DNA remains in the nucleus all the time. As we mentioned before, RNA has 4 bases, A, U, C, and G. So the one that's different between these two types of nucleic acid is T in DNA has been swapped to U. RNA is single-stranded, and the type of sugar consists in the molecule is ribose sugar. Okay, moving on to proteins. So once again, please make sure you know how to draw the generic structure for all amino acid. So amino acid is, okay, let me just jump in here a little bit. Amino acid is the subunit that make up proteins. Okay, so here I'm going to draw a generic structure for all amino acids. So there are different versions of amino acid in our body, but this is the generic or general structure for all of them. Okay, now this C in the middle is central carbon. So attached to a central carbon, on the top you have a hydrogen molecule. On the bottom, you'll see I've written R. So R is called R group, or sometimes referred as the functional group of amino acid. So this is the variable part that distinguish amino acid from each other. Okay, so that's what makes them different. On the left-hand side of our central carbon, you'll find an amine group. Okay, so there's N attached to 2H. This is a amino group. And then on the right-hand side to our central carbon, you will see a carboxyl group. So C double bond O and OH, this is a carboxyl group. Okay, so the only variable part will be R group. All the other different groups remain the same amongst different amino acids. Okay, now we know that protein is a complex molecule, so it consists of many smaller subunits of amino acid. They link together via a very special type of bond called peptide bond. Okay, and when they are linked together, we call this chain of amino acid polypeptide chain. Now, polypeptide chain is not the same as proteins because proteins need to be folded into a specific 3D shape in order to give them specificity and also make them functional. Okay, now I'm gonna just briefly talk about peptide bond. So let's have a look at this diagram here. Okay. So if you think about the general structure that we just studied before, you'll find, okay, so this is a central carbon, got one H. This looks like an R group. This is carboxyl. And on the right here, that looks like a amino group. Okay, so hopefully you'll be able to identify the different groups that's attached to our central carbon. So as you see here, as one amino acid come close to another amino acid, so this is another amino acid, the one of the H in the amino group will combine with the OH in the carboxyl group of another amino acid. And these two will join together and form water. So this is also referred to as a condensation reaction because H2O, that's water. So anything that produces water is condensation. Now, once these two molecules leave the amino acid they belong to, now the two amino acids become linked together. And this newly formed link between N and the C is called the peptide link. Okay, and also called peptide bond. Okay, so for all amino acids to join together, they need to have a peptide bond between the N and the C. Okay, now onto this kind of complicated looking picture on the right here. So this one. Okay, so you probably be able to realize that is talking about protein structure. Okay, for protein structure, there is a 4-level hierarchy. Okay, and once again, is not created with naming. So starting with level 1, the primary. So primary structure of protein. When we're mentioning primary structure of proteins, we are referring to the linear sequence of amino acid that make up a polypeptide chain. So the sequence of amino acid is determined by genetic code. Okay, so remember the previous slide, we mentioned that DNA code for proteins. This is the code. Okay, so the code that is contained in the DNA determines the sequence of amino acid that will be joining together in the polypeptide chain. All right, moving on to the next level, which is one level higher. So we're moving from primary to secondary now. So secondary structure of proteins refers to the local spatial arrangement of the polypeptide chain. If you want to give more detail, you can include the backbone. Okay, so backbone pretty much just refers to the constant part of the amino acid. So when they interact, that forms the secondary structure of the protein. You also need to know the common structures of secondary structure. And we are referring to alpha helix and beta pleated sheet. Okay, it's really important that you can identify them visually as well. So think about a helix, you'll probably be looking at something like this, and then pleated sheet is more like a parallel kind of structure. Oh, that disappeared. Okay. Almost there. Tertiary structure, I mean, halfway there. So tertiary structure of proteins is the overall 3D structure of a single polypeptide chain. This 3D structure is formed by interactions between the R groups, so the variable groups. So at this level, our polypeptide chain is finally functional now. So you can start calling it a protein. So a lot of the protein actually stop at tertiary level, but some of the proteins can go on and have quaternary level. So if a protein have quaternary level or structure, it contains multiple polypeptide subunits. So quaternary structure of protein refers to the 3D arrangement of multiple polypeptide subunit. Alrighty. So switching back to our slides. Let's have a look at a question together. All right, so this question asks, what is the secondary structure of a protein in the following pictures here? Okay, well, let's have a look. We'll have a look at each one of them. I know the answer is very obvious, but it's still worth to look at some of the other options. Now, have a look at the first one. Okay, that looks like a very complicated chemical structure, but let's try to interpret what we're looking at. Now, I'm looking at reading from left to right. This looks like an amino group in a polypeptide, in an amino acid. Amino group. How do I know that? Because I see one nitrogen with some hydrogen attached to it. And I also know the carbon that's attached to nitrogen will be my central carbon. Central carbon always have one hydrogen attached to it and also an R group. So this long chain of CH2 and CH3 looks like the R group for this amino acid. I also know a carboxyl group is attached to my central carbon. And here it looks like it has already formed a peptide link with the nitrogen from the next amino acid. So this link here is peptide link. Okay. And very quickly, I think I found some patterns. So the peptide link always exists between the COO in the carboxyl group of the previous amino acid and the nitrogen in the amino group of the next amino acid, right? So COO and N here. So pretty much I can just skim through the rest of the structure and try to identify that particular combination. So I found one here, COO with the nitrogen. So this bond, this dash between the C and N, that must be a peptide link. Keep doing that. COO and N, another peptide bond, COO and N, and that is the end of it. Okay, what am I looking at here? I have a chain of amino acid linked together by peptide bond and nothing else. So this must be a primary structure. Because remember, primary structure is the sequence of amino acid linked together by peptide bond, forming a polypeptide chain. Okay, now B. Okay, not much information is given by B except for a shape, and that looks like kind of a spiral shape that we drew before, right? So this, to me, looks like a alpha helix, and I know that alpha helix is part of the secondary structure. Okay, so very likely B is going to be our answer here. So I'm just going to go ahead and circle B, but I'll still have a look at C and D. Okay, so C is looking like a snake. It looks like a very long chain of polypeptide chain, and it's sort of like folded so that the molecule is 3D. So that, to me, looks like a 3D shape of single, because it looks like it's continuous polypeptide chain. Okay, and that belongs to tertiary structure. Okay, so it can't be C. What about D? Well, if you don't recognize these three-letter code kind of thing, these are the acronym for different amino acid. Okay, you don't need to worry about those acronyms. It's not required by our curriculum, but just know it looks like a necklace or a bracelet or chain of amino acid, which translate to polypeptide chain. So once again, this is another example of primary structure because it's a sequence of amino acid joined together by polypeptide bond, which means every single line between two amino acid units here is a polypeptide, sorry, not poly, just peptide link or bond. All right, moving on to question 2. This looks like a definition question. So the proteome is, well, let's have a look at option A. The total DNA content that is present within one cell of an organism. That can't be right because total DNA content is the genome. Okay, so a lot of times by doing some multiple choice questions that test your definitions, you can actually revisit a lot of the other definitions for like other terminologies by looking at the other options. So here, if you want to learn the definitions for genome, that will be option A. Okay, Option B, a complete set of chromosomes found inside of a cell of an organism. That is also incorrect because we know complete set of chromosome should be called karyotype. Okay, now option C, the entire set of protein expressed by an organism at a given time. That looks like the right answer. Okay, so that is the definition of proteome, the entire set of protein expressed by an organism at a given time. It's incorrect to say proteome is all the proteins that an organism can produce. Okay, so that is the incorrect definition. It has to be at that given time, what are all the proteins expressed by an organism? Okay, and finally, the four hierarchical levels of protein structure. No, because we already learned that the four levels are primary, secondary, tertiary, and quaternary, and it has got nothing to do with proteome. Okay. Question 3. Right. So if you're looking at a question like this without looking at the rest of the options and all that, you should immediately recognize this is a general structure of a nucleotide. Okay. And then with this knowledge, let's have a look at the question. So the question says a single DNA nucleotide is shown by subunits, X alone, X and Y, X and Z. So let's have a look at what each one stand for. Now, this looks a bit rotated, so a bit different to the one that we draw on the previous slide. And that is because DNA molecule is double-stranded and the two strands run anti-parallel, which means, okay, we have one strand, so the strand on the left-hand side here, that looks like the right orientation. But the other strand, in order for them to bond together, need to go the opposite direction. So this strand, or every nucleotide in this strand, they are rotated 180 degrees. Okay? But if your brain can rotate it back and see like the different component, just have a look at the one that's to the left. That would be probably the structure that you're more used to. Okay, now let's label it. X is this gray shaded area, and it looks like it's forming a bond with the other gray shaded area of another nucleotide. So the only structure that's in a nucleotide that's able to form bond with another strand will be the nitrogenous bases. Okay, so nitrogenous bases, they form complementary bonding. So base A always bond with T, C always bond with G. Okay, so X is nitrogenous bases. What about Y? Well, Y is the ribose sugar. So last but not least, we have Z. Z is the phosphate group. Oh. Okay, and this bond between the phosphate group of the nucleotide on the top and the nucleotide acid and the ribose sugar on the nucleotide in the middle here is also a good clue that we have labeled it right, because that is where the two nucleotides on the same chain will be linked, and they form these phospho sugar backbone. Okay, so the question is, what is a single nucleotide made of? Well, then it has to be X, Y, and Z, because that is the three component of any nucleotide. All right, moving on to question 4. Worded question. Please make sure you underline the keywords. So let's have a read of the question together. It says part of the mouse immunoglobulin that looks like a protein. Okay, and it bonds to an antigen is shown in the diagram below. 2 arrows pointed to 2 different types of secondary structure of this immunoglobulin molecule. Okay. It's also color-coded, even though it's in black and white, but we can see there is a darker gray heavy chain, like chain of polypeptide, and also a lighter chain, that's coated with lighter gray color. Question 1 asks us, give the name of each structure in the boxes provided. So let's have a look at the structure. We already know it's 2 different types of secondary structure. So you can choose between your options of alpha helix, beta pleated sheet, and random coil. So let's have a look at this circled region here first. It looks like the chain has got these parallel direction or orientation. And that is a typical structure of beta pleated sheet. The bottom one here, this twisted leather or helical shape, that is a good sign that this area has a alpha helix structure. Okay, now second part of the question, immunoglobulin also displays a tertiary structure, so 3D structure of single polypeptide chain, and a quaternary structure, 3D arrangement of multiple polypeptide chain. So referring to the diagram, explain what quaternary mean. And this question has got 2 marks allocated. So it looks like 1 mark goes to definition of quaternary structure. And the other mark, the second mark, you have to refer back to the diagram. And please make sure you be really specific when you're referring back to the diagram. Use the evidence that's actually given by the diagram. So it says heavy and light chain. You can even talk about the color coding, refers or suggests that there are multiple chains interacting. Okay, and that fits the description of a quaternary structure. So I'll let you have a go at that question yourself. And let's move on. Okay, now this part of the revision lecture talks about cellular transportation. Okay, you can have a read at the description yourself, but basically that is the definition of exocytosis and endocytosis. What I like to highlight to you is that upon exocytosis, so when vesicles leave the cell and goes to the extracellular region, the cellular membrane gets slightly larger. As you can see here, the membrane that makes up the vesicle fused with the cellular plasma membrane. So that added more phospholipid to the plasma membrane. So that's why we see cellular membrane get slightly larger, and vice versa for endocytosis. So as cells takes in molecules to intracellular region, so a part of the plasma membrane pinches in and enters the intracellular region of the cell. So that's going to make the cellular membrane get slightly smaller. Okay, so remember exocytosis and endocytosis are both involved in bulk transport. So bulk means you are transporting large amount of something. And this process is an active transport process. So that requires ATP. So it requires energy. All right, so here again, just some definitions. So please note that all internal transport within the cell requires vesicles, okay? And vesicles, it's made by a double layer of phospholipid, and that is exactly the same molecule that makes up the cellular plasma membrane. So it's also double layered. Okay. There's actually a lot of information in this diagram to the left here, but don't get too caught up in that. That's more Unit 1-2 bio knowledge. For now, you just need to know that AR help to synthesize lipids and proteins. It package it transport the proteins and lipids synthesized in vesicles and is sent it off to a Golgi apparatus. Okay, so these kind of parallel telephone looking thing, that's Golgi apparatus. The main role for Golgi apparatus is to store, modify, and secrete the vesicle. Okay, so please make sure you're able to distinguish between the functions and roles of AR and Golgi apparatus. So AR is for internal transportation. It also helps with synthesizing lipids and proteins because there's a lot of ribosomes and all sort of things attached to it. But Golgi apparatus is mainly for modification, storage, and secretion. Okay. So moving on to questions. Question 5. The diagram shows below represents a human cell secreting protein molecule. Okay, so this circular looking structure with little gaps, that is probably the nuclear envelope. And we always find AR attached or being really close to the nuclear envelope. And I'd like you to think about why that's the case. Okay, think about what's in the nucleus envelope. The nucleus, right. So in nucleus, DNAs are stored, and DNA has the code to make proteins. So it's really handy for ER with ribosomes. Okay, so these little circular looking things, they are the ribosome, to be close to the nucleus, right? Because once the DNA can be transcribed and carried out by mRNA, they get to the ribosomes and ER very quickly because they're really close, and start the translating process to make the code into proteins. Okay, now this parallel looking structure, so with strands parallel next to each other, is the Golgi apparatus. If you can't remember Golgi apparatus, just remember Golgi body. It's the same. And then it's done something and looks like it's made vesicles. And then the vesicles eventually undergo exocytosis and releasing the molecule, protein in this case, outside the cell. Okay, now question asks, the role of organelle Z, so this one, the Golgi apparatus, is to A, produce energy for protein production. No, producing energy is the job of mitochondria. Okay, so anytime you see producing energy, mitochondria. Transport proteins through the cytosol. If you see transportation, that is the job of AR because AR is kind of like tunnels, and it allows molecules and like newly synthesized molecules to get transported throughout the cell. Okay, so that's the job of AR. C, synthesizing protein for secretion. If you see synthesized protein, that is equivalent to ribosome. Okay, so that leaves us, sorry, I just dropped my pen. That'll leave us with option D, package protein into vesicle. Package modification, that is screaming Golgi apparatus. And that looks like D is our right option. Just on the side note, it's really useful if you know some of the general structures or visual representations of a few of the important organelles. Like this is a typical drawing of mitochondria. You'll see like an oval shape with cristae, that curly whirly line inside of it. AR is usually these like complex stacks of weird shapes. Sometimes it has got little circles attached to it. That's ribosomes. And if you see that, that is RAF AR. If there's no little circles, that will be a smooth AR. And the little circles are ribosome. Okay, moving on to 6. This is a 3D picture of Golgi apparatus. Because hopefully you'll be able to see some parallel stacks Golgi apparatus. These organelles would synthesize most of the ATP molecule. Okay, ATP synthesize, that's energy, so that's a job of mitochondria. B, modified package. Modified package, that sounds like Golgi apparatus. So I reckon the answer will be B, but I still will keep reading C and D. It says transport protein. Okay, transport, ER. And finally, D, assemble amino acid into proteins. So basically making proteins, and that is the job of ribosome. So almost identical to the question before, just asking a slightly different way. But the option hopefully is pretty straightforward to you, and that is B. Okay, I reckon I'll stop it here. It's been a pretty long video already. Next one, I'll talk about transcription and translation.